Welcome to day 3 of the virtual component of NeurIPS 2022. And today we're going to be having a workshop around the competition that actually all the folks on camera here helped organize, part of Open Problems in Single-Cell Analysis, a multimodal single-cell data integration competition across time and donors. A little bit of background, and then we're going to sort of get into presentations from the speaker. So one of the reasons that we're really excited about working with single cell data as a substrate for machine learning is that it provides sort of an unbiased description of cellular state. So think about cells as having billions of bases of DNA and hundreds of thousands of mRNA transcripts. And when we want to understand what they're doing, historically, people had to look at all of the sort of different cells in a tissue summed together. This is like taking a bunch of fruits and blending them into a smoothie and then trying to figure out, from this slurry, some properties of a kiwi, which is going to be very difficult. But in single cell genomics, what we can do is actually analyze every component individually. So we can look at all the different kinds of each type of cell as individual entities and then start to understand them that way. And the way that this has been really sort of enabled in the past 7 or 8 years is through the development of these novel microfluidic devices that take beads and individual cells and encapsulate them in this emulsion that you can then use to generate sequencing data. So this is measurements of the DNA, RNA, or proteins in individual cells. Just as these technologies have been developing, there's also been an increase in the development of algorithms to analyze the data. So when we were preparing this presentation about a month ago, there were 1,300 single cell analysis tools. And this is just going back to really the birth of single cell RNA sequencing in around 2015. So we can see this is actually both a very young field and it's growing pretty rapidly. And one of the challenges with such a explosion in the number of different algorithms is the need to do benchmarking. So right now you can find dozens of different benchmarking papers for different kinds of single cell methods. And one of the general challenges here is figuring out, how do we do this in a way that's fair and unbiased and continuously updated? One of the things that we noticed, surveying benchmarking literature. So I don't know how many folks here attended. There was a data-centric AI talk last Thursday as one of the keynote speeches at the in-person event. But benchmarking is incredibly important. And it's also really clear that when you do benchmarking just through individual papers, you can skew trends because everyone wants to publish and increase their next latest improvement over a particular benchmark, but that when you take sort of the same methods from these papers and then benchmark them in a standardized manner, this is like, run on a data set, processed in the same way, training for the same amount of time, using routine hyperparameter selection, we maybe don't actually have the kind of improvements that you might think. And so in 2020, those of us who was mentioning on the call, so Angela, Malta, Chris, more recently Peter, have been involved with organizing this effort called Open Problems in Single-Cell Analysis, where the goal is to do centralized benchmarking, aggregation of data sets, and host community events like this competition to sort of improve or fuel innovation in the world of single-cell analysis. And one of the things we're really excited about is this interdisciplinary community that we're building, where it's not just folks who have been doing single-cell analysis their whole lives or whole academic career, but also bringing in people who have a very different set of expertise to work on these sets of problems. I'm maybe just in the interest of time to get into the presentations, going to skip a little bit over some of this motivation and talk just about the competition that we have today. So for those of you who are just joining us, the competition that we set up uses multimodal single-cell RNA sequencing data that measures, each technology measures 2 of 3 measurements of genetic state. So it could be either the accessibility of different regions of the DNA. So we have for all of the genome, this measure of accessibility, the idea being that if DNA is open or closed, it's more amenable to expression. And then from open regions, and we also measure in the exact same cell another feature like the abundance of mRNA transcripts. So we know that there's some biological process that goes from DNA to RNA, and that this accessibility plays a role in it, but learning the rules by which accessibility leads to different levels of RNA expression is an open problem in the field. Similarly, we might measure the RNA content of a cell and a collection of cell surface proteins that are not shown here, and try to understand the rules by which RNA is translated into protein. And the way we set up this competition, we wanted to score, given sort of one input, we want to train on a particular target modality, and then we're going to score based off of correlation. I know correlation is not necessarily the easiest metric to train against in a machine learning competition, but we found based off of our competition last year that this would be a little bit more fair because different donors may have biases in the absolute value of a particular gene expression, but that the relative concentrations are what we really care about. And so we think that correlation is going to be more robust for this year. The data set that we generated was this multimodal time course. So we took hematopoietic stem cells, cultured them over 10 days, and then broke up the data in two different ways. So in one, we generate these multi-omic and site-seq libraries. We measure these two different modalities. And one of the splits we had, the public split in the competition, we had people predict across donors. And then in the test set, we asked people to make these predictions across days. So in site-seq, we gave days 2, 3, and 4 and asked people to make predictions at day 7. In multiome, we gave people days up to 10 for the ATAC component, the chromatin accessibility, and asked them to predict RNA at day 10. And the goal here is to say we want methods that both generalize across individuals and also that can predict across time, which is a much more complicated problem. This is sort of just a more visual depiction of this, right? We have this RNA, we want to predict towards protein. And so we want to know what are the relationships at day 7 into this test set. We were incredibly happy with the amount of participation that we had in this year's competition. So the final numbers were 1,220 teams. We had 1,600 competitors make almost 30,000 entries in the competition. And 84 different countries around the world were represented, which I think is really exciting to see. And as a lot of people who were involved with the competition know, there was also some shakeup as a result of this domain shift. So we had some people, so actually I meant to credit this. There's one community member on Kaggle who contributed these plots. But we can see that there are some people who did much better in the private leaderboard than the public leaderboard, including Suji Suzuki, our first place winner. We also have some real variance here where we have some people who are doing, actually, this is much better in private than public, and some people who are doing much better in public than private. And so we actually today invited both people sort of at the top of each of the private and public leaderboards, as well as some people who fell into the category of doing much better in private than they did in the public competition, in the public leaderboard, with the idea that this might lead to some better understanding of the time generalization, which is a very challenging problem. And finally, before I hand things off to Chris, I just want to mention too that a lot of this competition was fought over a relatively small amount of score improvement. I think this was something that we were really interested to see, was how much different the top competitors were. And we can see that even among just the top 20 people, the top set of algorithms all had relatively similar correlation. And one of the things we're going to look into next is like, even though the correlation is about the same, are there some that are particularly good at particular gene sets or in different kinds of cells? Chris, do you want to unmute yourself and
Sure, thanks, Dan. I keep it very short because we would love to start already with the solution presentations. So we also sent out the survey where we want to now understand a bit better what types of models actually perform well. Also, we would love to hear a bit more about the background of competitors. And first of all, I just want to basically support this point that Dan made, that we actually reached a very, very wide, and we're happy to see people coming from different fields, from like classical like technology and life science fields, as well as people that were already very, very experienced on Kaggle. Some were complete new joiners. And yeah, same with experience in analyzing single cell data. Then if you go next, we also see that represented in how prior biological knowledge was actually integrated in the models, and we're still collecting responses, so to actually get a more comprehensive picture. But what you can see at the very top there is the ranks, and at the like down below you can see which models actually integrated external reference in one or the other way, like pathway databases, or also whether people actually leveraged cell types labels, which is basically also that also includes basically prior knowledge on cell types that we expect. And yeah, let's maybe go already to the next last slide, which is now very much the work that comes now as a big follow-up, is what types of model actually performed well. You can already see, as you probably can guess, a lot of deep neural networks or deep learning models were used and performed very well. There were some like most popular loss functions that were also somewhat related to the evaluation metric. The cross-validation strategy was like, yeah, usually across donors and time points. And the dimensionality reduction, that one is, for example, a bit more interesting, which is like a classic single value composition, whereby the single cell field actually very often leverages a bit more complex methods like autoencoders. And yeah, just as a like the very last most important factors that competitors so far gave us feedback on is, of course, model architecture and the couple parameter selection, as well as the cross-validation scheme, which is now going to be very interesting to hear about more in detail from the winning teams. And yeah, the biggest limitation was hardware resources, but I guess this is kind of just due to the large data set. I would like to finish with the last slide, and we are like now very, very heavily working on the follow-up work. We're still collecting like server responses. We would be super grateful if you actually are, if there are listeners that haven't filled out the survey, wonderful. Yeah, so could do that. And yeah, then I'm very, very happy to now hear about the winning solutions.
Great, thank you so much, Chris. So just, we'll just get right into the talks. So Shuji, I think I saw you on the in the meeting. Are you here?
Yeah, I'm here.
Okay, I'm going to stop sharing my screen and let you present. Thank you so much for joining us, and congratulations on winning our competition.
Yes, thank you. Okay, so okay, can you see my slide? Oh, okay. Okay, let's start. Hello, everyone. I'm Shuji Suzuki, a data scientist at Preferred Networks. And today I would like to talk about my solution for this competition. First, thank you for the opportunity to explain my solution today. And overall, thanks the organizers of this great competition. And I'm interested in gaining experience in single-cell RNA-seq data analysis. Therefore, this competition was a good experience for single-cell data analysis for me. And first, I will skip this slide because competition overview was explained in the previous session. So first, I would like to discuss the difficulties I've faced during the competition. The first is high dimension data. For multi-ohm data, the input dimension is about 200,000, and the target dimension is about 20,000. Moreover, for statistic data, the input dimension is about 20,000, whereas the target dimension is 140. I believe these dimensions are higher than that of the data in other tabular data analysis. And the second is data noise. A well-known noise is a missing value in RNA-seq data. Particularly when the library size is small, the data have many missing values, I think. Furthermore, it is generally considered that there is a batch effect in the genomic data. So to address these difficulties, I focus on improving pre-processing in this competition. The model used is described in this section. The first is a Multiom model. There are input preprocessing, model, and target preprocessing as the main processing flow. In this presentation, I will only explain the essential parts. The first is Multiom input preprocessing. The important thing here is a binary input. It is also introduced in other team solutions. I use the binary input and notice an improvement in the performance. Dimensional compression is also important. I use truncated SVD in scikit-learn for dimensional compression. The output dimension was 128 I used. Next is target preprocessing. Here, preprocessing calculates the row-wise non-zero median at the expression level and divided the data by the median. After dividing the data by the median, I use a TSVD-based imputation method to address the missing value before dimension compression. This imputation method is dimensionally compressed once with TSVD and then transform the data back to its original space. Subsequently, it fills in the place where the value was originally 0 with the transformed value. At first, I tried scikit-learn's KNN imputation. However, it took long to calculate. Therefore, I choose this method because it can be calculated faster. Finally, I use TSVD for dimensional compression in this preprocessing. Next, I will explain the model. The model uses the data created by these input and target preprocessing. The model used in this competition is MLP I used. It consists two parts, an encoder and decoder. Both the encoder and decoder consist of linear layers and linear blocks. A linear block consists of GLU, layer normalization, dropout, and linear layer. The output dimension of the linear layer was 2048. The decoder outputs two types of data. These outputs were used in post-processing. The model calculates multiple prediction in this. During inference, the model calculates the average of each prediction to obtain the final prediction. This slide shows the post-processing of Multiom data. First, the model calculates the MLC loss with one of the outputs of the decoder and the output results of the target preprocessing. Next, the output the decoder is returned to the original dimension space. The correlation loss is calculated with this return value and RNA expression level data. This return value is used to calculate the next post-processing. In the next post-processing, MLC loss and correlation loss were used. The target data of MLC loss was calculated from RNA expression level data and the previous post-processing step. I calculated the final prediction value using the the previous post-processing value and the second output data of the decoder. So next, I will explain why I use such post-processing with multi-ohm data. The library size normalization is often used for single-cell RNA-seq data. It is also used in this competition. However, there is a large gap between 0 and non-zero value in the non-zero normalized data of this competition. This gap occurs a large error with compressed values using TSVD back to the original dimension. This figure shows the scatter plot of the original value and transformed value with TSVD. Clearly, the parts shown in red has a large error. To reduce this error, I use two techniques. The first was dividing count data by row-wise non-zero median value. It is used in the target preprocessing. The second was predicting regional target data and using it in computing the final prediction. It is used in post-processing. Next, I will explain the model of SiteSec. This is also has input processing, model, and target preprocessing at the main processing flow. I will also introduce important parts in this presentation. First is input preprocessing. Dividing by the median was used in this preprocessing. It is also used in multi-ohm target preprocessing. Afterwards, I selected the expression level data of essential genes as model input. Next, I used TSVD-based imputation method to address missing value. Subsequently, the data was dimensionally compressed by TSVD. It is an important part of how to select essential genes. Thus, I will explain in the detail. There exists an overfitting problem if we only select genes with high correlation coefficient between all training data inputs and targets. So I calculated the correlation coefficient for each batch and selected only the genes that was correlated with the target in many batches. Additionally, I selected genes from only related genes on the pathway. I use Reactome as a pathway database. When calculated the correlation coefficient was calculated using only non-zero value that do not have noise, I think. I use the Spearman correlation coefficient. It is robust even when outliers are included in data. Next, I will explain the target preprocessing for SiteSec. The preprocessing of the target data is more straightforward than that of multi-ohm. The important point in preprocessing is dimension compression. I use TSVD to compress this the dimension. And as a model for SiteSec, I use a model of linear layers and blocks as in multi-ohm. The decoder for SiteSec outputs one type data because regional target values were not used in SiteSec data. Post-processing of SiteSec was simpler than that of multi-ohm. The difference point is the loss function. I use MAE loss instead of MAC loss because the score of the model with MAE loss was higher than that of the with MAC loss. Okay, next I will explain the summary of key points. For chromatin accessibility, the binary input is important, and dividing by row-wise non-zero median value and imputing data was essential for RNA expression levels. Further, in multi-ohm, combined result prediction was important. And finally, in subset, using the expression level data of genes related to target data is important. Okay, next I will explain the local evaluation. I performed two types of evaluation in this competition. The main evaluation was cross-validation. This is a 5 cross-validation with data grouped by donor and day. The second was the evaluation for hyperparameter search. 5-fold cross-validation requires a total of 5 models training and evaluations. It requires more computation time. Therefore, I use this evaluation for the hyperparameter search so that it can be done in the short run. This is simple. I divide the entire training data into 2 data sets, 80% for training and 20% for validation. This table shows the local cross-validation score, public score, and private score for each of the important techniques I used. The baseline indicates the model without these techniques. Clearly from the table, the score gradually increased for every techniques. Okay, finally, I will explain how to make a final submission. I used the weighted average of the prediction for this model. One is first one is for models trained with changing the random set, and second one is models fine-tuned on only some batches. As a result, the final submission achieved a public score of 0.814 and private score of 0.775. Okay, this is the final slide. In this competition, I focus on pre-processing to tackle the difficulties of this competition. The key points are here. The source code is I released the source code, this URL. Thank you for the attention. Does anyone have questions?
Thank you so much. I think one quick question I was curious about, for the RNA expression normalization, you're doing this on the raw data, or are you doing this on the pre-processed input that we provided? Looks like it's the raw count on the next slide.
Yes, I used in this slide I want to explain the simply. So I write raw count data, but I originally in my solution I use library size normalization data and divide and compute same data.
Okay, and in your final model, you used the raw count data?
Yeah.
Okay. We do have a little bit of time for more questions. The next talk is going to be recorded and it's less than 20 minutes. So if anyone else has a question they'd like to raise. All right, great. Thank you so much, Shuji. And again, congratulations. Oh, we have one hand up, Malta.
Hey, thanks a lot. Really cool talk. I'm wondering if you considered using the time aspect of the data beyond your cross-validation scheme. So you mentioned that you're doing cross-validation by donor and day. Did you try to take into account the order of time points at all when making your predictions?
Please repeat the question.
Did you take into account the order of time points in your training data? Because you have information on day one, etc. And did you consider that?
So I use day this data day 2 or this data day 3. But I think I only use data.
Okay, so you didn't take into account that one day is before another day.
Yeah.
Okay, thank you.
All right, great. If there are no more questions, again, congratulations, Shuji, and we'll go to the next talk.
Thank you.
So we had to rearrange some of the scheduling. So I think the presentation order is going to be a little bit mixed up today. So the next presenter this is going to be a recorded presentation, and this is from our third place winner, Makoto Hayoto. I will just let's see. I'm going to make sure that the audio. Okay, so you should, when I hit play, one of the people on video just like nod if you can hear the.
Okay, hello, my name is Makotsu. Thank you for giving me the opportunity to introduce my solution today. I would like to introduce the third place solution of the multimodal single-cell integration competition. Since I'm not good at English, so I'm sorry if it's difficult for you to understand my English. Okay, let's go. First of all, let me briefly introduce myself. I'm working as a data scientist in the Japanese data analytics company. I'm usually engaged in e-commerce and developing recommendation systems. Before participating in this competition, I had no particular knowledge or experience in the bioinformatics field. The reason why I participated in this competition is that I wanted to gain experience in handling data that is different from what I usually use in my work. So I took this competition even though I had no experience in this field. Okay, let me begin by describing my approach to this competition. I will talk on the premise that do you know about this competition. First, after this competition started, I began to study the bioinformatics field. I had never heard of central dogma before. Fortunately, I could find a lot of information about it in the Kaggle discussions. I also read up the solution from similar competitions from last year to get a general idea of what methods I used. In particular, Alexander's discussion contained a lot of very useful information. If he is listening to this presentation, I'd like to thank him. Thank you. Okay, now let's move on. Let's move on to specific approaches. First, let me explain about the process of marginal data. For the chromatin accessibility data, many participants took TF-IDF. After that, they used the dimensionality reduction method, such as SVD. For me, firstly, I used the Okapi BM25 instead of TF-IDF because TF-IDF tends to produce less accurate results when gene expression levels are different per cell. To cover this drawback, I used Okapi BM25. Also, instead of using the SVD dimension reduction, I used the LSI, latent semantic indexing. LSI is one of the techniques used to condense a bag of words detail, and it works well in this case. This is implemented in the neural library that the competition host used for pre-processing. Let's the documentation for this library and notice that LSI is implemented. Next, for the site detail, I applied the same pre-processing that was done by the host. I normalized the expression amount in each cell and then took the logarithm. Okay, let me talk about the features I created. First, margin detail. The feature that worked best was one which transformed into 0 or 1 binary format and simply reduced to 16 dimensions with SVD. I also created the word-to-back feature. I created a string of genes sorted by the expression levels and condensed it to 16 dimensions using the word-to-back technique. This feature contributes very little to the public leaderboard accuracy, but when I checked the private leaderboard accuracy at the end of the competition, one-to-back feature contributes to it to some extent. Next, let's look at the site detail. Genes with protein names contributed well as features. It was introduced early in the public notebook on Kaggle. Also, genes clustering feature works well. I took the average of genes expression in each cluster and then reduced dimension with SVD. I used a gradient clustering algorithm, which is often used for network structure detail. For more information about clusters, the most commonly used method is k-means, which calculates the distance from the center of gravity of a cluster. However, when I tried to make same feature using k-means, the accuracy was dropped. From this, I understood the usefulness of the network-based cluster approach. And then, similar to Martinom, there were also features using back-to-back. This feature improves the public leaderboard accuracy somewhat, but it didn't contribute to the private leaderboard's accuracy. Okay. Next, I'm going to briefly share my approaches about the model and ensemble. There was nothing special. I built both Martinom and site models with simple MLP and CatBoost. The MLP is simply constructed with a combination of 4 layers. The Martinom had a target of about 23,000 dimensions, so it was reduced to 128 dimensions by SVD, and the loss is MSA, mean squared error. Site had 140 dimensions, so I didn't reduce it in particular, and I made collision coefficient loss, which is the metric of this competition. This loss was introduced in Kaggle public notebook, so I used that. Okay. For the ensemble, I built many models with different feature patterns, dimensionality reduction patterns, and so on. Martinom uses about 30 models, and site uses about 20 models. The final predictions were outputted as a weighted average. Weights are determined based on out-of-fold results. One of the key aspects of this competition was validation. I used a technique called adversarial validation. This is a technique to make the validation data resemble private test data. First of all, as shown in point 1 on this slide, I built a model to classify the private test data or the training data. Then, as shown in point 2 on this slide, the training data that is similar to the test data is used as the validation data. This approach worked well for this competition. In this competition, I built a classification model with LightGBM because LightGBM was more accurate than other classification algorithms. Then I selected 2,000 cells that were close to the test data for each donor and used them as validation data. If I hadn't taken this approach and I had decided that the public-based submission is my final submission, my ranking would have dropped to sixth places. So this approach worked well, I think. Okay, this slide presents the final submission and the score leading up to it. This table shows the approaches are taken for each model and the histories of validation accuracy and public scores up to the final submission. For Martinom, as I have discussed, the preprocessing and binary features contributed well, and the final score is greatly improved by assembling many models. For sight, the features of the genes associated with the protein works well, and model ensembles greatly improve the score. Okay, that is my all approaches in this competition. Okay, finally, I'd like to share some points that I found interesting. The first is that MLP was effective on biological data like this competition. I have always thought that gradient boosting methods such as LightGBM are more effective for table data competition like this. But in this competition, deep learning methods worked well. The characteristics of biological data and may fit deep learning methods, I guess. Second, features related to graph structure and order, such as latent clustering and what to bake, contribute to certain degree of accuracy. Further examination of features such as the structure of graph network and the order of expression levels may help us understand more about the gene's mechanism. I also implemented the graph neural network this competition, but since I didn't examine it very much, and I didn't have enough time to develop a graph neural network structure, so it didn't work well. Then in this competition, MLP was more accurate. Okay, these are my efforts in this competition. This competition was great opportunity for me to deepen my understanding of the bioinformatics field. This competition data is also very interesting, and I feel that there is a lot of room for trial and error with cutting-edge methods. If there is another similar competition, I'd like to participate in it. Okay, And I'd like to thank all those involved in this competition and those who listen today for this precious learning opportunity. Thank you so much for your kind attention. And thank you. Thank you so much. Bye-bye.
Okay, great. I think Makoto could not join us. And so we're just going to go straight into, there's going to be one other recorded presentation, and then we will go back into having some live talks. So just 1 second. Okay, this next presentation is from our 5th place private leaderboard winner, Lucky Shake.
Hi everyone, I'm from the team Lucky Shake, and my team won the 5th place in the competition. My name is Xiaodong Chen. I'm an undergraduate from China. I have been learning machine learning for half a year. I found that I'm very interested in it and plan to continue my research on machine learning and deep learning as the master's or doctor's degree. I have participated in several competitions on Kaggle, and I found that I'm more interested in tabular competitions. I will continue to learn on Kaggle. Before I begin to introduce our solutions, I would like to thank the organizers of the competition. They held such an interesting competition and gave us a chance to share our solutions. In addition, I have learned a lot from other competitors. Salute their open source spirit. Now let me introduce our general solution. We used the negative correlation loss to train MLP and used the root mean square error to train boosting model. In terms of CV strategy, we found splitting by donor seems beneficial for public LB, so we guess splitting by date for private LB may be helpful. Finally, we used splitting by donor and day. We used a large ensemble for side and multi, and this makes us from the 70th in the public LB to the 5th in the private LB. For side, we used the PCA to reduce dimensions, and the dimensions of the PCA is 512. We selected the first 75 currents and dropped out the following currents. We used the correlation and a greedy filtering method to filter important features. This greedy method is to add high correlation currents to the model. If CV is better, we will return them. Finally, we used the low standardization, which really improved the CV. We also tried some methods, such as IVIS, a recently proposed dimensions reduction method for biological macromolecules, and fake label. We tried to use it to filter some noisy features, but they didn't work. The ensemble for side came from 4 NN models and 2 HBM models. We used different activation functions and different pre-processing to train the 4 NN models and used them for ensemble. As for HBM models, we used different pre-processing and optuna for parameter tuning, which play a great role in the ensemble. Finally, I have a question. I tried to use 1D CNN in the competition, but its performance is very similar to MLP, and the correlation of their results is even higher than that of MLP with the same structure. I don't know whether such correlation is normal. Perhaps correlation can distinguish different models well. I hope to get an answer. Next, let my teammates continue.
So following the part about side stack prediction, I will now talk about our part on multi-ohm, which also was a really important part of the competition, of course, since we had to predict both. So my name is Jeroen Carpentier. I'm from Belgium, and I've been on Kaggle since August 2022, so around 4 or 5 months now. And for my education, I got a Bachelor of Science in Mathematics at Ghent University in 2018. And then 2 years later, in 2020, I got my Master's of Science in Mathematics focus on applied mathematics and informatics, also at Ghent University, where I did my thesis on causal inference and panel data. I'm also currently working as a PhD researcher in engineering technology at KU Leuven in Belgium, where I'm mainly focusing on computer vision on micro devices, so for example, like a Raspberry Pi. Also in computational physics, which I'm also teaching as an assistant, and also some electrical engineering, so actually manufacturing some computer vision systems in practice. Okay, so following my introduction, we can now talk about the pre-processing for the multi-ohm part. As many have done, and as we also did for the SciTech part, we did cross-validation based on day and donor. This was because we want to take the private and the public leaderboard into account, because we know that public was also only based on a new donor, but private was going to be based on a new donor on a new day. And we wanted our models to be robust enough for that, and that's how we incorporated it in our cross-validation strategy. This was also done in a public notebook, which obviously inspired us to do the approach in this way. So then the other part of the pre-processing, the actual pre-processing, the feature selection, it was actually based partly on the last top solution from the previous competition. Following that pre-processing, then we generate 4 neural networks, which are then finally combined to generate the final prediction. So I'll now go deeper into that. As I said, our cross-validation strategy was then followed up by the feature selection, which was based on a previous solution. We start off by using a TF-IDF, or a term frequency times inverse document frequency transformer, which was then followed by just a simple log1p transformation. And then consequently, we did an L2 or a max normalization, but in our final solution, we ended up using L2 normalization, then just principal component analysis, and after that, we run a row normalizer, so that's per cell ID on each row. This feature selection was actually then combined with just a normal raw principal component analysis without any extra augmentations or anything. So based on that, we choose the 100 most important components from the more advanced technique on the left, and then we chose the 64 most important components from the more basic PCA, which were then concatenated into a vector of 164 input features. And these are all the same for the 4 neural networks that we ended up using in our final solution for multi-ohm. So following that pre-processing, I will now very briefly go over the models that we used. So we had 2 base linear models with only linear layers, and then we had 2 models that use convolutions. So this is the first basic model where we had the GLU activation function. This was decided based on our cross-validation. So we did cross-validation. Based on that, we choose the different or like the best activation functions to use in the final model. That's our first model. It has around 17.5 million parameters. So the amount of parameters is way higher for multi-ohm than for SciTech. Then the second model is basically the same model as the previous one, where we switched the activation to a swish function. So based on our cross-validation, we saw that these 2 were the best candidates as an activation function within this structure. Then our third model actually is a convolutional model, which has 15.7 million parameters. So as you can see, the trainable parameters are around the same as the linear model. It has a CELO activation function, and it is finished in the end. Before a flattened layer, we have a max pool layer, which ended up being pretty important. Based on cross-validation, we saw that the impact of this layer was pretty big. And then our last model is basically the same model as the other convolution one I just showed, but it has a hard switch as activation function, which is, if some are unfamiliar with this, it's basically a computationally less expensive variation of the normal swish, which replaces the sigmoid part of it. So having generated the 4 predictions using these 4 models, they were then combined just by basically ensembling them. So the mean of the 4 predictions, as you can see here, and before they were put together within the mean, they were standardized per cell ID, which was a method that was posted in a public notebook. And I guess many of the competitors used this approach to get the best ensemble. And then very briefly, some things that we tried also that didn't work. So we had initially only cross-validation on donor. We switched to day and donor only in the last couple of weeks of the competition. And as some know, getting a good cross-validation score on donor was pretty challenging. And I tried to push this by using the fairly recent model soups paper, which is basically just finding the best combination of weights of a model that's trained under different hyperparameters. Of course, it did push the cross-validation, which is pretty trivial, but it did not push the leaderboard, only in terms of multi-ohm. We tried it for SideSec, but it did boost the leaderboard a tiny little bit, but we did not continue using this because of the lack of time. It takes a lot of time to train on all these hyperparameters, and it keeps you restricted to just using that one model while you generate the new model. So it doesn't really add that much diversity to your final solution. So following that, I would like to thank you for your attention. I would also like to say thank you to Kaggle and the organizers of this competition. I had a lot of fun, only having joined within the last 5 weeks. Working hard on this competition was very fun because I think it was an interesting cause, it was interesting data, and we had some good competition. And of course, I also had a very nice team, so I would like to thank my 2 teammates. Thank you for your attention.
Great. I think we might have Jerome on the call, so I don't know if you'd like to introduce yourself. I see Jay Serpent, which I think is the, but
yes, I'm here. I don't know if you can hear me clearly.
Yes, we can hear you wonderfully. So I don't know if anyone has like a question or two before we go into the next presentation. Malta?
Hey, thanks a lot for a cool presentation. And I'm curious, you mentioned very briefly, I didn't catch all of it, the most important features that were used from combining the kind of 4-step pre-processing versus the PCA. I'm wondering why you decided to go for this kind of 2 different approaches to pre-processing and combining these, and if you looked into any kind of redundancy in the features that were pushed forward in this approach.
Yes, that's a good question, and thank you for your question. Well, basically, the way that we found this final solution was just by a lot of experiments through cross-validation. And so we did raw PCA, just some features. And then we also did a whole bunch of other methods, like in the public notebooks, for example, there were a ton of different pre-processing methods that were proposed there. And then we just found that, well, we tried all these different options, and then we decided to just try different combinations. So it's really just a matter of trial and error. And we just found out that using these 2 methods and combining, like concatenating these 2 methods actually work best for a cross-validation. And in the end, it also works best for the leaderboard.
Okay, thanks.
No problem.
Great. If there aren't any other questions, we have.
Oh, sorry. Hey. Thanks for the presentation. That was really interesting. I was wondering whether you could outline a bit whether how much your assembling helped. So your models seemed related, right? So they were all neural networks. Did you try out assembling only with the same model architecture? So just use different seeds to train your model and assembling that, and then trying out different architectures. I would just be curious, like to which extent assembling had boosted your performance.
Yes, okay, that's a good question. So basically, as to ensembling just the same model structure, we did not really try because, as I said in my presentation, I used the model soups method for that. So basically just generating a whole different set of weights and taking the best combination of that weights for one model structure. But it ended up being really time consuming because you need to try out all these different hyperparameters. So what we basically just did was we basically take the models that have the best single leaderboard score, and then we decide to combine these ones. And that's actually what turned out to be the best. So that's something that in general, I always like to do on Kaggle. When I'm trying to ensemble, I always look at the best single models, and then I look at combining these single models. What's the result from that? And usually there's always some kind of gain there.
Cool, thanks.
No problem.
All right, great. We have next is going to be our second place winner, Jinzan or Senkin. I don't know how you'd like to introduce yourself and get started with your presentation.
Okay. Let me share my screen. Do you see my screen?
Yes.
Okay. So I will talk about our second place solution. First, let me introduce my team. I'm thinking, basically, I don't have any biology knowledge or experience, but I'm active caregiver. I'm caregiver grandmaster. My teammate has some experience in biology. So we have a good combination with our strengths. So we have been the first place in public leaderboard for a long time, but finally we shifted down to second place. But we are satisfied. We have a robust result. And okay, let me skip some basic information. Let me share some highlights of our team. First, about pre-process. So I don't have some biology experience, so I needed to search or see the discussion topics. So I found one article of Nature introduced the normalization. First, I searched DSB transformation. Then I found another transformation normalization is popular. It's centered log ratio. So I tried this method and compared to others like lab size or lab size than log 1P or TF-IDF or some other. But I found the CLR is best. And I also see the last year competition solutions. I found the row-wise this square transformation was popular, and I also tried. Yes, it improve my model. It can improve. And my teammate, he has some experience in biology, so he created a combination of some methods commonly used in single-cell omics. So the flow, like first use the row count, then normalize the sample normalization by mean value over features. It means subtract the mean value of rows, each row, and then use squared transformation, then feature-wise this score, and the last is batch effect correction. So we think the most challenging part is the future information. It may cause domain shift. So he take a day as a batch. For each batch, calculate the column-wise median to get a median sample, represent the batch. Then subtract this sample from each sample in this batch. Well, we observed it didn't improve the score very much, but we think it is simple to avoid the overfitting. And we observed the PCA, the distribution is better. The different days distribution is better than without batch effect. Okay. And about feature engineering, I I use the CLR transformation, then use truncated SVD. Yeah, it's also found in last year's competition. Most competitors use truncated SVD. I found it's best in my model. I tried a different number of dimensions. I found 200 is best for site and 100 is best for multi. And we use high correlation features. We select the top correlated features both by day and donors, because we just select the highest correlation, it may cause overfitting. So we split by day and by donor and select what the top correlate features, and finally select the all the high correlated features in different days and different donors. And then my teammate used his pre-processing, then he tried a different PCA. Finally, he used 3 different kinds of PCA, PCA, IPCA, and factor analysis for side part. And my teammate also used the CL type one-hot encode. But the one-hot encode, it didn't improve my model. So it's a little, I think, because we start with the different neural network architectures or different parameters, so the features have a different impact. So we have some difference in feature and different model structures. About validation, I think the biggest challenge is how to build a robust model for unseen donors and unseen days, especially the days, because the private test is unseen days. So at an early stage, I used a random k-forward, because I think if I use the group k-forward by donors, it will not train all the train the model enough. So first I use random k-forward, and I found the local cross-validation score and the leaderboard score matched very well. So because the public test from unseen donor, so we can think we don't need to worry about donor domain shift. But the time domain shift is unpredictable. So after team merge, I found my teammate also use random cross-validation. So we have good cross-validation score and the leaderboard score, but that maybe also our mistake. We haven't optimized the out of day validation score. We checked our features one group by one group with out of day validation. It can make sure all the features can improve every day. Yeah, it's okay, and I think it makes our model is robust, but we haven't optimized the validation score for different days. So I think it cross we shake down at final private test. And about my model, I think I have many experience in tree model like LightGBM. So I think I use LightGBM get good performance because this is multi-labels. So we can use neural network to train one model, very simple, or we can train LightGBM model with 104 140 models for site. So my the overall architecture like here. So the left part is about my pre-processing and feature engineering I just mentioned. The right part, so I use LightGBM to predict, to train model and output 140 predictions, then use truncated SVD, then use row-wise Z-score. So the predictions of LightGBM will treated as an input to neural networks. So I first tried a blend with LightGBM and the neural networks. It only improve a little, but if I use this method, it's like stacking. Use LightGBM predictions to neural networks. It give me much more improvement. And I have two structures of neural networks. Basically, it's start from three hidden layer MLP, and I have some experience to transform the dense layer to GRU layer. For example, the first model is replace the first layer dense layer with by GRU, and then the second model I add one more by GRU layer after dense layer. It can improve a little compared to three dense layer, and some other parameters. I think the two models I use different loss function. One is cosine similarity, one is MSE. And the activation is also important. Basically, we start from LELU, but I found the target is DSB normalization. They have many negative values. So if we can use activation can have a negative value of hidden layer weights, it will better. So here I found ILU is better than LELU, and switch is also good. Also, one trick is I concatenate the three hidden layers at the last layer. Basically, we don't need to do this, but in my cosine similarity loss function model, the three hidden layers concatenate is better. And I also tried pseudo labeling. It can also improve a little, both in local CV and public leaderboard. Okay, the second neural network, I also use target Z-score. So it didn't change too much, but the brain diversity is good for ensemble. My teammate, TMP, he used 13 layers MLP. Yeah, it's a very deep. I never used such deeper MLP, and he used harbor loss. So our structure is very different. So our ensemble is very good. And about multi-part, the LightGBM is very hard to train because there are 23,000 dimensions. So first I use a PCA to transform to 1,000, then train 1,000 LightGBM models, and then inverse them to original dimensions. My neural networks also can do this. First use PCA transformed target, then inverse them. And also I can use only one model to predict the whole dimension directly. So the overall structure like this. So the TF-IDF transformation is good for traditional models like LightGBM, Cat Booster, Kernel Ridge, and Ridge. Yeah, this this is also like site part is use this model prediction, then input to neural networks. And the multi-part, I don't found much use for transformation. And finally, my teammate, the multi-model leaderboard is very low. So I finally just submit my multi-model. So I only use the centered log ratio for normalization. And I also have 2 neural networks, simple 3 hidden layer, dense layer, and cosine 2 different loss function, cosine similarity, and hub. And activation is a switch. The Gaussian dropout is a little bigger, 0.3. Other are not, no special. Okay. I think I'm finished. Thank you.
Thank you so much, Senken. Does anyone have any questions? I see 2 people with their hands raised. Maybe let's start with Malta.
I thought you were going to start with Peter because I've been asking everyone. But thank you so much for the very extensive talk. I noticed that so at the end you kind of mentioned your ensembling takes, for the multiome, takes 2 different pre-processing approaches, right? And first is takes TF-IDF, and the second thing has a more kind of I forgot, he's like, yeah, CLR, transform TSVD, etc. Why do you, so this kind of the assumption behind this is that there is no best pre-processing, but that you're going to use the ensembling with your cross-correlation approach to get the best possible output. Is there no kind of one way or one pre-processing approach you thought was better than the other, or why choose multiple different approaches here for pre-processing?
So my thought is how to find pre-processing is similar with target normalization. So in side part, the side part because the target is used DSP normalization. So I found, but I cannot use the DSP for the input pre-processing. So I use the similar CLR, central log ratio. That's my thought. Yes, I also tried many different pre-processing. We can use some libraries, Snappy or Moon. But yeah, we need to try and error. I select the best method.
Oh, sorry, did I misunderstood you? So you selected the best pre-processing and you did not use multiple pre-processings and ensembled over them. You just selected the best from all of these. Okay.
I just use the best.
Sorry, thank you.
Let's maybe go next to Alexander Churbov.
Hello, everybody, and thank you very much, Senken, for your talk, and thank you for the organizer. So I have a small question. So you was on the top of the leaderboard just from the beginning, and so we thought that you found something like a magic feature at the beginning, because just at the first week you're on the top. So we thought you have something secret solution, but it seems it's not correct. So can you maybe shed light on your initial models on your first approach, which I guess was not that much complicated as the final solution?
Okay. So my initial model, I start from LightGBM because I'm familiar with this kind of model. So here, and my initial model is just LightGBM, both site part and the multi-part. But then I found a neural network is better, and I tried to use the stacking to input LightGBM prediction to neural networks. It boosts very much, especially in leaderboard. So I have been first place in public for a long time because I used the LightGBM prediction as neural network features.
Don't you remember how much was the boost from LightGBM to the second step? How much it improved?
For example, when everyone is 0, 8, 1, 2, I was 0, 8, 1, 3. So I compared to other competitors, I think the difference is 0.001.
Okay, thank you very much.
Peter, do you have a question?
Sure. Thank you so much for the presentation and great job. I have a very brief question. I was just wondering whether you observed that adding cell type as a covariate improved your model.
Sorry, could you repeat?
So I've seen you said you included it as a one-hot encoding.
Oh yes.
And you would expect it to improve it. I've just seen some people including it, some people not including it, just like explicitly. I was just wondering whether that helped your performance.
Okay. Actually, it cannot improve my model, but my teammate use this one-hot encoder from initial model. So we know if we start from some features, after we cannot remove those features, initial features, it will make your model performance worse. It depends on your neural network structures or parameters. But yes, it includes useful information. But in my model, I think I have other strong signals information, so it cannot improve my model.
Thanks.
Great. Thank you so much, Senkin, for this wonderful presentation. And we're going to go next to Oliver Wang, who is our 4th place team winner.
Hi, can everyone see the PPT?
It looks great, but if you can go into, yeah, okay, perfect. Presentation.
Oh, okay.
Thank you.
Hi, everyone. I'm Goshen Wang from Tongji University, and I have just finished my undergraduate study, and I'm now working as a research assistant at Tongji University, and I'm also applying for my graduate study. And today I'm going to talk about my solution to this problem. And here is the background and the data, and since you may all be familiar with that, so we'll make it really quick. So we need one problem is to predict the gene expression, and another problem is to predict the protein levels. And first, I want to talk about the CV strategy. And according to the data spelled here, and we can see that the trained data set is 3 days and 3 donors, and we need to predict this 4th donor for the 3 days at the public test and predict the day 7 of the 4 donors. So we can make simple CV strategy, but since there are many there are many batch effects. Yeah, there are many batch effects. So we'd better do something like the data spells here. So I did a public like cross validation strategy here, and I use 2 donors for the training and 1 donor for the validation, and regardless of the days. And here is a private leaderboard like cross validation strategy here, and I use 2 donors for 2 days for the training and not using the 3rd donor, and use another day for the validation. So since that we can see the result of the private leaderboard, so my strategy was that I used this public leaderboard to find like the best model, the best model architecture, or the best parameters. And then I keep everything but the cross-validation strategy to change the cross-validation strategy to the private like ones and keep the other things and to find the best result of these two and use them as the final submissions. But actually, I found that the public like cross-validation strategy is better than the private like ones. I haven't found the real reason for this, but I guess it's maybe because my model, I assembled a lot of model, maybe because this one out of luck, or maybe I should use a time series cross-validation strategy here. I will try to figure out later. And next is the side part, and here is I use the raw data, and here are 3 parts of my input features: the dimension reduction features, the important feature, and the category features. And then I put it into the model and get the results and use correlation laws and add them with L2 regulation to train the model. And for the dimension reduction features, I used TSVD for the linear decomposition and use UMAP for the manifold decomposition. And I also adopt Novel's method of last year's competition. And I was meant to replace TSVD by this method, but I found that the public leaderboard score and CV score worse if I just keep Novel's method. But if I keep both of them, the score will grow a little bit. So finally, I keep both of these 2 features. And next, for the important features, the name important features is the same as Andrew M's notebooks here, and I add some additional gene information from the MyGene package. And for the correlation important features, I chose the top 3 features that correlate with each target and their overlaps. So the final dimension is about 104. And for the random forest important features here, I chose the top 128 important feature of the random forest model. And after dimension reduction, it found that there seem to be aggregation of different cells, different type of cells. So cell types may be also a relatively important features. So I will next give a brief introduction about the gated map neural network. It's a network that's designed by myself, and I will show the process of the designing. So here is a simple MLP, and we have features here and get predicts here. And the cell could be just a simple linear and the active function. So if we want to get better scores, we may do something like that. And we concat the result of these cells and use MLP to make the predictions. And this model is also one of the best model on the public notebooks. And next, I want to get more improved. So I found that this is kind of a one direction flow. And what if we turn it into 2 directions? So I made something like that and found that this really improved the cross-validation result and both the leaderboards. So can we do more? Yeah. So I add some other inputs here to make more process of the data and plus the data transform here to get the data to input, and here becomes the backbone of my model. And next, I want to improve more, so I did something to the cell here. So I replaced the simple MLP to a structure like here for the cell. And I use the linear dropout, linear active function, but not active function after this linear. And I found that this architecture can better deal with the overfitting than just put a mesh here. And it's better than just one linear and mesh. So I found this, and I also add dropout here. And so can we do more? And I was fascinated by the transformers. So I want to do something like transformer. Actually, I tried the architecture like the transformer, such as RTDL or the other architecture, other models. So, but they all failed. They don't score higher than the simple MLP, and maybe the reason comes from the feature vectors. So I was wondering if I can use something that don't need feature vectors. So I add something like that, and I called it the gated MLP. So because this softmax here and the index transform here is something like the attention mechanism, and it don't need to be feature vectors. You can just put in data here, do the transform, and put it out here. And so actually, this is something more like the gates in RNN, such as LSTM or GRU. So I call it gated MLP. So if this works, it's hard to say. I did improve in the CV score and the public leaderboard, but it's worse the result in the private leaderboard. So it's hard to say this really works. And so I did some alumni experiments after the competition. I replaced this one to the same MLP like that. I called it no gates, and I used the one direction model here and call it no map. And I reduced the inputs here and call it no inputs. And here comes the final results. And the original model topped the public leaderboard here, and the model with no gates a little bit lower than the original model on cross-validation result and public leaderboard, but it's a little bit higher in the private leaderboard. And another thing I call it no input is ranked the top of the cross-validation results, but it's much lower in the leaderboard. So maybe we can't trust much about the cross-validation strategy. And next part, I'm going to talk about the stacking method. Here is a screenshot from a course offered by Professor Mu Li, and the left is the model architecture about his multi-layer stacking method, and the right part, he talked about how to deal with overfitting in stacking. So he the first approach used less data. So he introduced the repeated k-fold bagging for the overfitting problem. But in this competition, we cannot just do simple k-fold bagging, because, as I said before, the special cross-validation strategy can improve the results. So we need to combine the k-fold bagging with the k-fold group by donor or by day or etc. So I do something like that. It's the public-like cross-validation strategy I told before, and I spelled all days into 3 parts, and regardless of their days, just random spells, and randomly use these parts, randomly spell them into 3 parts, both the train and the validation, and use this for the training, use this for the validation, and use another for the out-of-forward predictions. And here is the full process of the training process and the prediction process. I add the original feature here as the video shows here, and the results turn out that the stacking is the best on the CV score, and it's also the best on the public leaderboard. And next, I also trained some other models here. And if anyone is interested in this model, you may check my code on GitHub. And I did some example in the end. And next is the multi-part. I did nothing very special here. Just here, I directly did a dot product here to get to transform the output of the model to the predictions. Since we did some dimension reduction here, and we can use this part to turn it back, and we don't do the correlation loss on this part. We can just use the correlation loss on the predictions, and then the grades can directly transform from this part to the whole neural network. So we are not optimizing the dimension reduction result. We are actually optimizing the final result. And then I train some other models and do the sample too. And that's all for my talking here. And thank you. You may get more information on my GitHub or the discussion. Thank you.
Wonderful. Thank you so much, Oliver. We're a little bit behind schedule. And so I think what we should do is we have one recording to play. And then Fernando, I see you're on the line. Will you be okay presenting 10 minutes behind schedule? Is that okay?
Yeah, that's fine.
Okay, all right. So we're just gonna go straight to the studio, and then we will go to continue on.
Hello everyone, my name is Yuri and I am a member of Team Chromosome, which took the 7th place in Multimodal Open Cell Integration Challenge on Kaggle. And now I'm gonna present our team's solution. Since the principle by which test data is divided into public and private parts was announced from the very beginning, we decided to take advantage of this and build our local cross-validation as similar as possible to the private part. In other words, it was important for the model to be generalized to new donors as well as to days from the future. At the same time, the generalization to new days had more weight since only one new donor was appearing in the private part of the test data set, but the day was exclusively new. The diagram here shows the validation scheme we have chosen for the side-sec part. For the multi-ohm part, we used the same validation. The only change was the number of folds. They were 9 folds instead of 6, since we have one additional training day. Towards the end of the competition, we slightly modified the scheme. In fact, the only change was that the validation fold now included observations from the one nearest day from the future. Let's go through every step in both problems, starting with side-sec data pre-processing and feature engineering part. Each of our team members had their own approach to data pre-processing, so we had several data sets using different pre-processings, what generally increased the diversity of our models for fine ensemble. And here are the main points we have done. First of all, we have removed features with constant values. The second one, we found some important features from raw ones. Various approaches have been used to find such features. It is worth mentioning 2 of them. The first one was to find top 200 features for each target by the absolute correlation values. Afterwards, 300 features were selected by occurrence frequency from those tops 200. And the second approach was to get default feature importances from the single multi-output PyBoost model. We will talk about this model a little later. But this approach was very simple and fast to calculate, and was also able to find non-linear relationships in the data, unlike selection by correlation coefficients. In reality, both of these methods produced fairly similar feature subsets. For features that were not included in the list of important ones, a PCA was used to reduce the dimension, and we also generated aggregates of important features at the donor day level. Such features did not work for neural networks, but for our GBDT model, it increased mean cross-validation score by approximately by 3/10,000. Thus, the final trained dataset was a concatenation of the important features without any preprocessing, as well as the principal components of the dimensionality reduced features. Unlike the SciSec part, in the multi-ohm task we had just one variant of data preprocessing, and the total number of features was 485. Main points here: we have removed features with constant values too. Also, we have applied PCA on the entire training dataset, plus we have applied PCA on groups of columns with same prefixes in their column names. Here I mean that, for instance, we have features which names start from CHR16, CHR17, and so on, and for each such group we have trained its own PCA. And the key thing here is that only the last available day was used for training. So our final models were trained only on data from day 7, and days 2, 3, and 4 were dropped from training procedure. This approach raised our local cross-validation score by about 2-3,000. And what models were helping in achieving that result? Our final solution for SciSec part was an ensemble of 5 models: 3 fully connected neural networks, 1 convolutional neural network, and 1 gradient boosting model. In our case, it was PyBoost model. The weights in the blend were selected with help of Optuna, and all our neural networks used correlation loss. PyBoost model used standard mean squared error loss, and some other things which were used. Neural networks used skip connections, and 3 neural networks of 4 used pseudo labels. PyBoost model used target splitting into random groups. It improved our local cross-validation score by about 4/10,000. And in the end, I should say that PyBoost model was our best single model for SciSec part. And just a few more words about PyBoost. This is a new implementation of gradient boosting on decision trees, and it supports multi-output tasks out of the box. It is very fast in comparison with multi-output CatBoost, for example, as it utilizes GPU. An article about this algorithm was published on NeurIPS this year. So if you are interested in theoretical foundations of this algorithm, all links will be provided in the end of this presentation. If we are talking about multi-ohm part, the final solution was an ensemble of just 3 models: 1 fully connected neural network, 1 TabNet, and 1 PyBoost model. The same loss functions were used here, and some other details: that neural networks were trained on all targets, but PyBoost was trained. But for the PyBoost model, we reduced the dimension of the dataset with targets to 64 components with PCA. This was necessary because the GPU available to us didn't have enough video memory to store such huge targets matrix. And PyBoost was our best single model again and here. And we also consider it's very important to mention things that did not work for us. So let's start with SciSec part. Classic bottleneck autoencoder. I should admit here also CatBoost with a multi-RMSE loss. During our local experiments, it had approximately the same score on cross-validation as our worst neural networks in the ensemble. However, since multi-RMSE is only available on the CPU, the training was extremely long, and in the end we dropped this model. Also, some alternative losses were tested for PyBoost, such as mean absolute error and Huber losses. And we have tested some variations of mix-up for neural networks, also without any positive result. Also we have tested splitting targets not into random groups but into clusters for our PyBoost model. But unfortunately, the result was not positive for us. And stacking with PyBoost as meta model also worked worse than usual linear blending. For the multi-ohm task, also some alternative losses for PyBoost were tested. Also, we have tested zero inflated models, which can potentially work better with a very sparse target matrix. Also, unsupervised pre-training for TabNet was examined, but again without any positive result, and some other tricks like skip connections for neural networks, also without positive feedback. And the last thing we have tested is feature selection of important columns, important raw features with PyBoost and CatBoost models, but it did not work for us too. So here are some useful links to dig into the PyBoost if you would be interested. And thank you once again. Feel free to contact me via Kaggle or email, and goodbye.
Okay, great. So we're just going to go straight into the next presentation. So Fernando, would you like to take it away?
Hi, can you hear me well?
It sounds great.
Okay, let me share my screen. Let me know if you can see it. Can you see my screen?
Yes,
we can see it.
Okay, thank you very much. My name is Fernando. I'm known in Kaggle as Via Lactea. First, I would like to start by thanking the organizers for giving me the opportunity to present to you my solution today. If I go over my time, please feel free to interrupt me. I'll try to stick to the 10 minutes. Okay, so let me move to the next one. So when a competition ends in Kaggle, the question that's in everybody's mind is, what's the magic? Meaning, what is the technique or approach that gave the top teams an edge? And everyone is striving to find the magic. In my case, when I started on the competition, I thought I could find the magic dust in two things. One was about increasing the amount of training data. I mean, usually all else being equal, the more data you have, the better models you're going to end up with. I tried to look for other sources of data that I could use. I couldn't find any. And what I thought about doing is the test data had 70% of the cells in use. So I thought about using it, labeling it with pseudo labels, and then use it as part of my training. The other idea I had was using NLP transformers. And then this is because the way I understand the biological processes behind this competition, essentially you're doing transcriptions from one form to another, say from RNA to proteins. And NLP transformers do an excellent job at that with natural languages. So I thought about using them. And what I intended to do is take the original input. This is the pre-processed data, and most of it was zeros. So I would binarize it and then assign a token ID to each of the columns, and then use that as my input sequence to the model. Now, this then I could either produce directly the target values, or I could use a sequence-to-sequence model where I would binarize the targets, and again, I would try to predict those binarized ones. And the reasoning behind that is I looked initially at the correlation between the actual targets and the binarized form, and the score was pretty high. So if I could do a good job predicting binarized targets, I would be doing a good job with the actual targets. Now, the challenge with, or one of the benefits of doing this, obviously, was the data was very large. In the case of the multi-home section, I couldn't really train with all of it. I would need to use some sort of reduction. And there's always some data loss with that reduction. And another way of doing it is using what transformers do. And the assumption there was that it's more important than the actual value of each of the input values was whether it was present or not, whether it had, it was 0 or something else. And if we, by using this type of transformation, we would ensure that we would not lose any information other than, of course, the binarization. So it had that benefit as well. Obviously, it was challenging because you would, even with the amount of data resources that I had, which is the Kaggle servers, you would not be able to train with the whole data. You would have to break it in some way. And one of the ways I thought about doing was say, for example, for site sequence by chromosome, build a model per chromosome. But even then, that would not be enough because the chromosomes, you would have still more, you would end up with sequences larger than the usual transformers can handle. So I would have to further break it. And because of that complexity, what I decided to do is leave this to the end. And this was my plan when the competition started. So first, work out my CV strategy, my feature engineering, because those are things I did not want to change later on, because they would generate rework. Then train models, ensemble them, generate pseudo labels, and repeat this a few times until I had a good set of pseudo labels. And then with that, having my final set of data, work on the NLP transformers. Unfortunately, as I finished as I got my first batch of pseudo labels and I tried to train with them, what I found is that it was degrading considerably my scores. And at the time, I wasn't sure whether it was because the data on the private leaderboard, it was different than what we had, and that was what was triggering the worst score, or if it just wasn't working. I gave it another try. I tried to improve the models to see if I could come up with better pseudo labels, see if that helped. It didn't, so I ended up dropping the idea. And by the time I got to the NLP transformers, it was too late in the competition, and I didn't have time to make them work. I again run into out-of-memory issues, and literally I was hours away. This was the night before the competition ended, and after many sleepless nights, I just realized that I wasn't going to be able to try it. So I'm very curious to know. I didn't notice that anybody else tried NLP transformers. I don't know if anybody outside of the competition has ever tried them. I'm certainly curious to know whether they would have worked or not. Now, about what I did, my CV strategy, as other presenters mentioned, it was a bit challenging because we had a very small domain. In a typical Kaggle competition, you would have had, hundreds or thousands of days and donors. In this case, we had a small set, and you had to make a prediction for 3 of the donors on an unseen day, and for the other donor, it was unseen both the day and the donor. And one of the things that was concerning to me is when I looked, for example, at the multi-home data and the scoring of my models, the day 7, they performed significantly worse. And the concern is, well, is that gonna be the same with site sequence on day 7? And what about the day 10 of the multi-home? So to tackle that, what I tried to do is mimic the situation with the prediction we had to do. So for these 3 donors, we had to predict for an unseen day. So I picked for the validation one of the days, and I trained with the other ones. As for the 4th donor, what I did is I picked a day and a donor for the validation, and then trained on the remaining. And this is how my submission would look like with my D and D for day, DD for day donor approach. So I would use a combination. I never cared about the public leaderboard because it was substantially different, and I had this concern with the day, the time change. So, but one of the concerns I had here was I was using just a small fraction of the data for training. So because we can use 2 submissions to hedge my bet, I used this DD approach where I would train with all the data available outside of my validation. This is how they performed on the different scores, and the DD approach, so that extended the last one, performed better across the board, significantly better. Feature engineering, I did what a lot of other folks did. For site sequence, I used both the original data, and this was the pre-processed data. I never tried to use the raw data. I didn't think it was worth the time. I also added the type of cell, the gender of the donor, the day, and also the mean for donor day for each of the columns, the number of non-zeros. Tried a few other things, binarized data, the SVD of the binarized data, SVD for the means for the neighbors didn't quite work out. Areas I grayed out are the ones that didn't work. I tried the original target binarized, the SVD of the target, something similar to multi-home. I tried combinations of the targets with multiple loss functions and weighted each of them. That didn't quite work out. Models, I tried a number of them. All my final ensembles were based on residual networks. This is the simplest one I've used, pretty small. But if you notice, the scoring on the CV was pretty close to my final ensemble. And this is my final solution, linear regression of 16 residual networks for the sequence. Same thing, but with 13 networks for multi-home. Tried a number of other things. Ultimately, I think what gave me, I couldn't find any magic really. And in the absence of that, how I explain having gotten one of the top positions is my feature engineering gave me a boost over just the data that was provided. He also provided diversity, which helped with the ensembling, which gave me an additional boost. I ended up with several good models. And then I believe my CV strategy and the way I used it helped me ensure consistency between my CV results and the private leaderboard, and allowed me to end in one of the top positions. And I think I'm a little bit over my 10 minutes. Thank you.
No problem. Thank you so much. How much did you find that the encoding the cell type and day and donor information improved model performance?
They did. My best models, typically they used, so I tried multiple combinations. My best models used the normalized means and the normalized counts of non-zeros, as well as type. And so that was my best models, but I tried to have diversity, so I didn't stick to just the things that provided better performance. I tried a number of things, and then I just let the ensemble figure out which ones were best. But yeah, they performed. When I looked at individual scoring, they helped. They gave me a boost. And one of the things I did, for example, my best multi-home models use day only as additional feature. But I didn't use that on my final ensembles because I noticed that this was for DDE, because on the other approaches, they performed worse. And I wasn't sure if I wasn't overfitting. So I disregarded some of my best combinations of features.
Thank you. Malta?
Thanks for the really cool talk. I think it's a pity you didn't get a chance to get the transformer working, given that you already have such a boost towards the private leaderboard. Maybe there was a lot more potential there as well. I'm curious how you encoded the metadata that you added. So day specifically, did you do this as a kind of one hot, or is this ordinal still? Did you use the actual day? Because if that seems to be, this would be kind of the only model that we've seen so far that is using some kind of ordering of the data as input to predicting a final output. I'd be curious.
I used the day as is. The way I thought about this computation, this was, I mean, in an ideal world, you would have data for the same cell over time. I think because of the way this is measured, it's impossible. But that would be ideal in my mind. I tried to use, for example, RRN models and LSTM. And unfortunately, we didn't have enough data. If we had data for multiple days, even from different cells, I thought we could use that. I made an attempt of using this. I don't have time to go into details, but at the end of the day, what helped most was just using the ordinal value of day. My best models on both site sequence and multi-home were using that feature.
You did test other ways of encoding day as well, and this worked best if you used ordinal?
No, I used this one. Well, yeah, I did. I made a beginning an attempt of using one-hot encoding, but this one just using the value worked best. I tried really a number of things that I didn't go over. Usually, what I would do is, especially in the beginning, is if it didn't boost my score, I would just drop it. Eventually might have helped with diversity, but there's so many things you can do with the time you have. This is an hobby for me, so I would just, if something didn't improve, I would just discard it.
Okay, it's really interesting from a biological perspective, looking at feature interactions between the ordinal day encoding and any of gene expressions would potentially give us quite a lot of information on what are genes that are actually changing over days. To see whether we can kind of compare this. So that's really interesting. Thank you very much for your talk.
Yeah, thank you.
Wonderful. Thank you so much, Fernando. We're going to move on to the next speaker, who is Ambrose. Ambrose, we can, I think we can hear a little bit of background noise, but we can't. Okay, now we can see you.
Okay, and you can hear me?
Yes.
And then I only need to show the presentation. Can you see the presentation?
We can see it perfectly. Thank you so much.
So I'm Ambrose Mazzetta, and I'm going to tell you about the lessons I learned during this competition. I'll start by giving an overview of the solution, and then I want to dive deeper into two specific aspects, the pre-processing in SiteSeq and the aspect of time in the multi-ohm part of the competition. So here you can see my complete pipeline for the SiteSeq model. If we begin at the left, I begin with the raw gene expression data. And then I have a pre-processing block with the library size normalization and the log1p. I'll dive deeper into that later. And for the pre-processed data, I do a singular value decomposition to reduce the dimensionality and to separate the signal from the noise. And in parallel, I select about 300 important columns from the data. And originally, I selected the important columns by name, but then I changed to a method where I computed the permutation feature importance and selected the columns with the highest permutation feature importance. From the metadata, I took only the cell type, and then I concatenated all these features and used them for basically 3 kinds of models. The first one is a neural network which optimizes the correlation loss. The second type of model is a neural network with the MSE loss. And with this network, with the MSE loss, I found that it was better not to predict the 140 proteins altogether, but to reduce the dimensionality of the output with a PCA from 140 to 90. And then, of course, you need the inverse transform to get back to 140. And the third model I used was kernel-rich regression with a RBF kernel. Kernel-rich regression has a quadratic memory complexity. So for this one didn't run on the Kaggle infrastructure. For this one, I needed the Saturn cloud. And here, apparently, it was better to predict only 60 components and then do the PCA transform to get the proteins. For the multi-ohm part, I start with the attack-seq data. I reduce the 200,000 dimensions to something around 100 with the singular value decomposition. And then I wanted to use the cell type as a feature. In the site-seq data, the cell type is given for every cell, so that was easy. In the multi-ohm data, we didn't get the cell type for all the cells, so I had to create a simple classifier which predicts the cell type so that I could add the one hot encoded cell type to the data. And then I created a k-nearest neighbors model. The interesting thing about k-nearest neighbors is that you can predict very many targets efficiently. So with k-nearest neighbors, you can predict 20,000 targets at once quite efficiently. And I think there aren't many other algorithms which can do that. It turned out that using 40 neighbors gave the best result. But then these 20,000 predictions, they contain a lot of noise, and we have to denoise this. And here it's useful to know or to guess that the data doesn't use all the 20,000 dimensions. In fact, if you reduce this to 35 dimensions, so then you can see that all the data points more or less live in a 35-dimensional hyperplane. So we can denoise this prediction by doing a PCA transformation of the prediction to 35 dimensions, then we transform it back. The other model is a neural network. And for the neural network, I took only the day from the metadata. And then you can use the day as a feature or for weighting the samples. I'll go into detail of this later to capture the time aspect. And then the neural network predicts around 100 dimensions of the output. And then with the inverse transform of a PCA or SVD, you can get the final predictions. So let's go back to site-seq and pre-processing. In the original data of the competitions, these RNA gene expression levels, they were given as library size normalized and log1p transformed counts. And this pre-processing, it distorts the data. You can see this in the diagram. In the experiment, we count molecules in a cell. And then we get an integer number, 0, 1, 3, 4, and so on. And the typical normalization multiplies this number by 100, more or less, and then takes the log1p. And then you see these orange dots. That's where these points are transformed to. And we see that the 0 and the 1, they are transformed to points which are quite far apart. And this is a pity because the raw counts, they are counts of molecules, and it's easy to count one more or one less. So if your raw count is 0 or 1, basically it says nothing. That's just noise. And with this transformation, we are amplifying the noise. So what I did, I compared different pre-processing methods. So in this experiment, I used different pre-processing methods and then always the same neural network to make the predictions. You see the results here. The longer bars are better. If we start with the raw data, then we get some medium score. If we do only the library size normalization, but no log1p, we normalize to 1 million, the score gets even worse in the second row. But if we choose another number than 1 million, so maybe 100,000, that's already better, or 10,000 is even better. So it seems at 10,000, normalizing to 10,000 and then taking the log1p gives a good result. Normalizing to 1,000 is worse. And just for comparison, I compared this with not using the log1p transformation, but the square root transformation, which I think was proposed by Senking and TMP. And the square root transformation is similar to log1p with a well-chosen library size. And maybe this pre-processing result, it's not the best one for every possible application, but it seems that for a singular value decomposition, it's good to look at the pre-processing, because if we have this large distance between the images of 0 and 1, this basically creates outliers, and the outliers have too high of an influence in the SVD. The other questions I want to look at is, what is our model of time? So in multi-ohm, I can either make a model and say, we have X, which is the DNA accessibility, from which I want to predict Y, the RNA levels, and this function is independent of time. Sure, we see that for days 2, 3, 4, and 7, maybe the data points, they don't have the same distribution, but we can imagine that the model, this black line, is valid for all days and can be extrapolated to day 10. Or is it a better model if we say the RNA levels, they are a function of the DNA accessibility and the day? If I try to show this in the diagram, this means we have the 4 days, and every day gives another model. For every day, we can fit another straight line, and then we can extrapolate this to day 10, which in this diagram would have a straight line far above. And I think this is a relevant question, because in the left model, we are saying that the cells live in the moment. They don't have a memory or a history. In the right model, the cell somehow knows knows the duration of the experiment. The cell knows which day it is. And for this, I did some experiments. First, I trained on the data of one day and predicted the data of another day. This gives a 4x4 matrix. Here we have the training days, we have the test days. And in this matrix, we see that the score is good if the training day is near the test day. So if we want to predict day 2, it's best to train on day 2 or maybe on day 3, but it's bad if we train on day 7 for predicting day 2. And vice versa, if we want to predict day 7, it's bad if we train on day 2. In the next experiment, I tried to predict day 7 with different methods. So I said, either I can oversample day 4, because day 4 is the nearest to day 7, or I can use the day as a feature. And if we look at the results here, the first bar is the baseline. I train on days 2, 3, and 4, all days with equal weight. The score improves if I oversample day 4 3 times. And the score improves even more if I oversample day 4 5 times, or if I drop the other days. In one of the presentations before, we heard that that team had the best result when they trained only on the last day. That's the same here. And then I compared this to the approach of using the day as a feature. And if you simply use the day, the integer number as a feature, 2, 3, 4, 7, then we get a bad result. And I think this is because extrapolating from when training in an interval from 2 to 4 and then extrapolating to 7, that jump is too large. And for this reason, I clipped the day to the interval 2 to 4.5. So this means days 2, 3, and 4, they are trained with value 2, 3, and 4. But for day 7, I give 4.5 as input to be in order to not be too far away. And for day 10, again, I give 4.5 as input. And with this feature, the result is even a little better than training on only day 4. So the conclusion of this is that the RNA levels, they do not depend only on the current attack-seq data, but as well on the cell's history. That's it. Thank you for listening.
Wonderful. Thank you so much, Ambrose. That was a wonderful presentation. Maybe Malta will take one question, and then we've got 3 more talks to get through before the end of the hour. So,
sorry. A really cool talk. Thanks very much. I'm wondering if you considered parametric models of time as well, because you mentioned about training on subsets of your data to improve kind of the history in there. Did you consider trying to parameterize this further?
Yes, my last 2 models, they had the day as a parameter. And the main point here is you cannot use the day as a parameter directly, but it may need some transformation of the day.
Okay.
And
thank you. Yeah,
in this transformation, it showed to be optimal if when I clipped the day, so that the 4 and the 7 weren't too far away.
Okay, I didn't catch that. Thank you.
okay, great. Again, thank you so much, Ambrose. We're going to go next to Tianyu. So the next two presenters we picked because they did especially better on the private leaderboard than public. So these are benefactors of the shakeup. So thank you so much, Tianyu. Please. I know we had originally scheduled 15 minutes. I don't know. I don't want to like rush you. Maybe if there's any introduction stuff, you might feel free to skip.
Okay, great. Yeah, thanks. Can you hear me, by the way? Yeah, thanks. Okay, so yeah, so many thanks Daniel for his for introduction, and thanks for the organizers again for holding such a great competition. And I'm Tianyu Liu on behalf of Team One Only. I will present the lessons we have learned from the second open problems in single-cell analysis competition. Yeah, so we jump a very high position based on switch between the public leaderboard and the private leaderboard. So I just go over these problems very quickly because in our model design, we actually pay attention to 3 difficulties obstacles existing in this problem. The first thing is about the time series existing in the data sets. Because we know that the cells are extracted based on some donors, and we perform the sequencing toward these cells. It's actually like a disconnected approach or discontinuous approach. This is a major difference between our current data with the traditional time series data. And the second thing is about the heterogeneity existing in different samples, because in our public test, we actually only need to evaluate the performance of our model towards 1 donor, but in our private data set, we need to judge it based on 4 donors. So another thing we need to consider about the difference existing in different donors. And the last thing is about the cell differentiation. So these 2 good figures is created based on the Amber's post. And we can see that for the cell proportion based on the time series, the proportion of HSC is becoming lower and lower. For other cell types, they are becoming higher and higher. So this is the problems caused by cell differentiation. And we hope that a good model can predict the tendency of such cell differentiation and simulate this process. Okay, so based on these 3 kind of ideas, we can start to introduce our models. So actually, our models contains 2 parts. The first thing is about pre-processing steps, it's including the feature selection. And the second one is the main body of our model. It includes the neural network and 3 kinds of tree-based boosting models. And finally, we apply the model ensembling towards our output. And cross-validation is also a very important approach here because we need to use CV scores to actually predict the performance of our model towards the private datasets. And we utilize the group threefold based on the donors because we need to consider about the heterogeneity existing in different samples. And we also utilize this kind of design to try to simulate the time series of the donors. And we also add a very simple 5 voice into our final submissions because we need to handle with the problems caused by the overfitting. So we believe that this approach can actually increase the stability of our model. And for the standard sequence part, we need to predict the protein expression based on gene expression. So here, the first step we use for the data engineering is to select some important genes based on the genes name overlap between the whole genes group and the predict proteins. And the rest of the genes, maybe around 84, I think. And for the whole single-cell sequence data set, we also perform the singular value decomposition and try to get the top principal components. I call this as principal components. And then combine the important genes with the SVD results for the top principal components as our final input. And then we will use this input to train our custom neural network. It's an MLP and 3 kinds of state-of-the-art gradient boosting decision tree-based models. And after we have the output of different models, we can try to combine them together based on the model ensembling. And we use weighted average approach here to generate our final submission file. And for the targets file, we directly predict the protein expression of each of these datasets. But for the multi-omic part, things has been changed because we still utilize the same model structure and the same cross-validation scheme. However, the single-cell ATAC sequence data, it is very sparse. So we need to perform the denoising approach before to input the single-cell ATAC sequence data set into our model. And we utilize the data binarization toward the single-cell ATAC sequence data set and then use SVD to perform the dimension reduction for the input part. And for the output part, because we have many genes we intend to predict, so we cannot directly perform to set the output of our model as the dimensions of the single-cell sequence data. So here we also perform the SVD based on this target sets. And we save the loading matrix after SVD based on the training data set. And this will be utilized in our future prediction. So now we can input the
our training files into our models, and then we will get the predict principal components here. So we will multiply the loadings we have in the training data set towards our predicted principal components. So here we can have our final cell by gene matrix. And finally, we perform the model ensembling here, also based on the weighted average method. And this method is known as a variation of the input and output data. So this approach is provided by average. We believe that to increase the stability of our model, we can try to adjust the dimensions of SVD operation towards the multi-omic part. For example, we can generate many SVD based on different dimensions like 60, 70, or 80. And for the output end, we can also try to adjust the dimensions of the SVD. So finally, we can combine all of them together based on the crossing scheme and to generate the many outputs and then average them into our final output here. So this is another types of the model ensembling approach, and it can really contribute to our score improvement here. And another interesting part we have found is about the training optimization process toward the neural network, because we need to select a suitable neural network with good structure toward this problem. And the first thing we have tried is about the residual network, and I know many participators use this tool. And we use residual network to prevent the gradient vanishment existing in the deep neural network training process. And the residual neural network means that, for example, in this simple 3 layers neural network, we will connect the output of layer L2 as a partial input of layer L. So it means that we can connect with the past information of layer 1 and with its future prediction part. So this approach can handle with the gradient management part in the training process. And the second thing we have tried is about the activation function. So we also need to find a suitable activation function to perform the no linear transformation between these 2 layers. So we look up some papers and we finally find that CELU is the best candidate here, because we know that according to organizers, the output of our models or the true predicted value, they're actually being standardized, which means for each cell, the means is 0 and the variance is 1. So CELU as an activation function, it can actually ensure that the output of our model will be converted to such a distribution with mean as 0 and variance as 1. And this process is known as a self-normalizing. So that's why we finally choose CELU as our final activation function. And the last thing we have considered about is about optimizer. So here we don't utilize the well-known ADAM and SGD with momentum, but we choose another type of state-of-the-art optimizers here, known as ADA belief. So according to the research related to the ADA belief generated by the author of such tool, under these 4 cases, ADA belief can actually generate a better estimation compared with other state-of-the-art optimizers here. It will be more close to the optimal point. And for the rest of parts, I intend to talk about some additional functions we have added towards our model. So this includes some file-driven methods here. The first thing we have tried is about the data penalization. I mentioned it previously. This approach is generated by Jianyao. And here we mean that for the cell by pick matrix, we can select each cell and then divide it by its maximum value. Then we perform the scaling operation here to transfer a cell by pick matrix into a 0-1 binarization matrix. And if we take a look at the heat map generated by the picks information and cell type label information, here we can see that we do not observe a huge difference in the heat map. But if we try to plot the cell type distributions based on UMaps for the training data set, we can see that before such operations, the cells are very massive and the cell types are mixed together. However, if we perform the data penalization after such process, we can see that we can observe a very clear borderlines existing in different cell type labels. So actually, this approach can handle with the noisy information existing in the single cell ATAC sequence data. And the second thing we have tried is about data imputation toward the single cell sequence data set. We know that since we intend to generate the cell by gene matrix, and for single cell sequence data set, it is very sparse. Some zeros existing in this matrix are generally true zeros or the true zero gene expression. But for other type of zeros, they are caused by the technical issues. So we choose some techniques that can help us deal with these issues or actually reduce the effect of such technical issues toward our prediction process. The first thing we have tried is metric. So this method is based on the diffusion map and the nearest neighbor information. And the second tool we have tried is about the deep impute. So deep impute is a kind of neural network based on the denoising autoencoder neural network based model towards data imputation. So we perform the output of these two tools and apply it based on the important genes we have selected previously. However, it is pity that we did not observe a obvious score improvement after the data imputation operation. And the second approach we have tried about submodal optimization, because if we choose to plot the correlation existing in the set sequence data set, the correlation for the predict value of the model output and the true value in the training data set, and we can see that for some proteins, they are very easy to predict. For example, the CD41 and CD71, the correlation is very, very high, nearly like 0.9. I believe that the reason is because they are very, very important in our body, in our cell, the functions of cell types. But for some other proteins, they are very difficult to predict. For example, the CD64 and the CD83, the correlation is even lower than 0.5. So this is very interesting, and I believe that is because their functions are not very as important as the previous proteins, and they lead to some uncertainty here. So to handle these things, also this problem, we utilize the old models of the neural network I mentioned previously to predict the proteins which are easy to predict. However, for other proteins, we create a new model based on neural network with new parameters and new training epochs and new strategies to see if we can improve the correlations for the rest of the proteins. However, we did not observe a very huge score improvement here. Yeah. And the last thing is about the metadata embedding. So metadata means that there are some extra information we apply to our prediction process. They may be contributed to our training prediction process other than the profile information. So the metadata embedding information, including the gender and the cell type label distribution and the days we perform the sequencing. So we utilize some embedding models to include all of such metadata information and then transfer them based on something models like we use the touch embedding here, but maybe we can also try some like word to vector or the large language model like transformer to generate the final vectors. We can combine these vectors with our input data here to finalize our input file, and we can use this input file to perform the model prediction. So we finally include such design into our model submission because we believe that this approach can actually increase the variation and the stability of our model. And in the rest of the part, I intend to discuss the lessons we have learned or the interesting phenomena we have found toward this competition. The first thing we have realized is about overfitting is very difficult to find out. Even if we plot the distributions of the input file, both for the training dataset and testing dataset, even if we did not observe a very obvious shift in such distribution, we still missed our best candidates, like over than the 0.772 in our final submission file. And I think the reasons may be very divergent. The first thing I can consider about is improper single-cell pre-processing steps. And Ambrose has a very interesting post related to this area because actually he proved that the data normalization and log1 transform are not the best approach to predict the perform the pre-processing steps for single-cell sequence dataset because it will increase the random noises in a very large scale. And the second thing is about the feature selection. So feature selection is also a very important topic here we have tried. So we only use the important genes and something based on the SVD, but we can also try some method based on the manual void preservation in the single cell area. But such methods are very time consuming. So maybe I think we can try this approach later. And for the last thing we can try is about the different loss functions. So we only use mean squared errors here. However, I believe that there are some other methods like cosine similarity or the huge loss that's used by other participators that we can try to replace the mean squared errors with this loss function to see the performance here. And in this slide, I intend to talk about some possible downstream analysis we can try in the future. In the left figure, this is proposed by Nancy Zhang's paper, and they also intend to predict the surface protein expression based on the gene expression. So here, they choose to analyze the results of model output based on the marker proteins for each cell type labels. They plot them and then observe the protein enrichment in each cluster. So they use this approach to prove the correctness of their model, and it is very illustrative. And the second thing is about the SIGNET. So this is a tool towards the correlation analysis between the chromosome and the gene expression. So because our model can actually catch the importance of the input features based on some kind of mechanism, we can compare the peaks we choose as important towards some genes and to compare it with the peak to gene linkage generated by the SIGNAC to see if our model is really the peaks we selected is really contributing to the prediction process for some genes. Okay, so to summarize my presentation today, so in the problems related to the time series, the cross-validation scheme needs to be chosen carefully. So we cannot only choose a simple k-fold, we should consider about the donors and the time series. And in the single-cell data related works, so for models based on the neural network can perform very well. So this is very surprising towards me because I think for some other tabular-based method in the Kaggle competition, nearly the tree-based model can outperform the neural network because the later one is very complicated. But in single-cell area, it seems that things have changed. And the model ensembling approach is a very, very, very effective solution to mitigate the overfitting. And the belt-driven methods are very diverse and do not perfectly fit the application task scenario, I think. And the last thing is, although we missed some unhappy things in the last few days of this competition, but I really enjoyed this game and learned many knowledge about the gap between the research area and the application area. And I think these are the most important things existing in this single-cell competition. And for the acknowledgement part, firstly, I intend to say thank you to my close teammates, Jian Zhou, he's a graduate student at Beijing Institute of Technology, and he proposed a data penalization approach. And Eric Grant, he's a data scientist in Germany, and he tried to utilize different SVD input and different SVD output towards the multi-omic part. And for me, I'm a PhD student at Yale University, advised by Professor Hongyu Zhao. And our lab is focusing on the research related to genetics, biostatistics, and bioinformatics. If you're interested into our research, you can scan this QR code to more information. And for the codes I used in this presentation and the whole competition, you can scan this QR code for more information if you are interested in. And this is a reference page, and many thanks for listening. And I'm happy to hearing some questions and comments. Thanks. I hope I do not use more time.
Let's see.
Oh,
are there any burning questions? Otherwise, we might just go straight. We have 2 more presentations in 20 minutes. And so we might want to just let's see, Hongzi.
Maybe I can use task to answer the questions if it's okay.
Yeah, absolutely. That's very much encouraged.
Yeah. And maybe you can continue.
The person who asked the last question is also our next presenter. Who actually, yeah, Hongzi is actually one of the winners of last year's competition.
Yeah, I know it's dance. Or maybe I can text to him and he can continue his presentation because of the time. Yeah, thanks.
Okay, thank you. So am I supposed to start my presentation?
Yeah.
Okay.
Maybe if we can, just in the interest of time, try to keep it like close to 10 minutes or so. I don't know if that's possible, but you know, if we go over.
I will try my best, but I think it could be hard because I think, yeah.
It's fine. Take your time.
I prepared for 15 minutes. Sorry. I'm Hong Zhiwen, a PhD student from Michigan State University. Today, I'm going to present the solution of our team DANCE, which is a graph-based seismic prediction model. It's a graph-based model, and we are ranked at 24 without ensemble because we don't have ensemble models. And today, I'm not going to talk about engineering stuff because I want to address our uniqueness. So I'm going to focus on our exploration about the graph-based models in single-cell analysis with our datasets. And then I will introduce the model we use and our new solution, what we modify to our last year's model. And lastly, I would also introduce a DANCE package, which we built it and we integrate our method into it. Okay, so first one, I would like to talk about the cell graphs, which is a very popular method in single-cell analysis. Because we assume that when we use the graph-based methods, we are actually introduce some extra denoise effect. For example, if we build a k-nearest neighbors graphs based on cell similarity, and we use like if we apply graph neural networks to this graph or any other graph-based method, we are implicitly solving the graph signal denoising problem, where we try to find a clean signal that is close to the original input and is smooth across the whole graph. Okay, so our journey starts from the empirical study of the cell-cell graphs on real-world dataset, which is the competition dataset. So we first have a feature transformation, which is MLP layers, and we add a feature propagation over the KN graphs we build before or after the transformation. So this kind of architecture called decoupled GNN. And we noticed that, unfortunately, this kind of GNN doesn't improve too much. So in some cases, it improved a little bit, but it's definitely not significant. So it makes us confused. So we make a lot of analysis, and finally we make two interesting observations. So first one is, if we choose a random cell and we plot the correlation based on the RNA Pearson correlation and the the protein Pearson correlations, basically it's a correlation between all the other cells and a random selected cells. You will see that if we connected the K nearest neighbor graph based on the RNA features, we are likely to connect the right part, those cells, to the target cells. But you will see that the protein correlation is not actually very slow. And there are actually some cells on the top side that are very similar to the target cells in the target space. However, we can not distinguish them from the input space. So that means when we build the graph based on the input, like based on the RNA-seq, we cannot build an accurate cell graph. So that's why this kind of graph-based method might not help. And also we make another observation that our prediction, the performance, is related to cell activity. So we use some existing software to calculate the cell activity scores, and we find that there are some cells that we predict really bad, and all of those cells has a larger cell activity scores. So what we learn from it is that first, to simply build a cell-cell graph or k-nearest neighbor graph might not help too much. The main bottleneck in this task might come from the uncertainty of cell activities. Basically, if the cell is actively evolved, it's very hard to predict. And we also noticed that the denoising technique plays a crucial role in this task, because we noticed that SVD actually provides a very strong baseline. And we also find that for SVD, if we select the first 4,000 PCs, it only covered about 58% of the variance. And even though we use top 75 PCs, it already achieved a very good performance. So it means that there are a lot of information that we directly dismissed before we train our models in the pre-processing step. I think SVD should definitely not be the optimal solution, because we lose too much information along with the noise. But it seems like we don't have a better choice to distinguish those less important information from the noise. That's why the SVD provides a very strong background, baseline, I mean. So this is what we learned from our first exploration. And next, I'm gonna introduce the SAMOGN, which is our last year's winning methods. And I will introduce how we adapt it to our new methods. So this is SAMOGN, where we first construct a bipartite graph based on the input matrix. So we convert the input matrix to a bipartite graph, to the adjacency matrix. And then we insert the networks, the gene networks, into the graph. For example, if we take the CySeq as an example, we have the cell gene graphs, and we also have a gene networks, and we insert the gene networks into the graph. And after construct the graph, we use a heterogeneous graph convolution layers, is a GNN, and we extract the cell representation, and eventually we use the MLP to do the prediction. And what we used for the gene networks last year is the hallmark gene sets, which is a pathway data set. It tells us the relations between genes. And last year, we also noticed the batch effect as the difference in equipment cell donors. But this year, it's more like the donors and the time. But last year, we designed a batch features which is we first calculate the statistical features of each cell and then aggregate the features of all the cells in the same batch to be the batch level features. But it seems like it doesn't improve too much to our models in this year's competition. Anyway, we first try to directly apply our last year model to this year's data set, but we found that there is a limitation of it. Because this year we have more than 22,000 genes, and last year it's only 15,000. And this year's sales number is also larger than last year. So we encounter a scalability issue. Basically, in the graph, the number of edges is much larger than last year. So it takes much longer training time than MLPs. And it means that the hyperparameters are very hard to tune for our models. So we can only use the default setting to train a model, and we find that it outperforms some baselines like the denoising autoencoder, or directly use the 4 genes to train an MLP, but it cannot outperform the SVD. So we continue to polish our idea and to try to improve the performance. And here comes our new solution. So we first replace the pathway hallmark gene sets with string, which is the most famous networks in single cell biology. Since string is actually a protein interaction network, so we not only have if we first map the string to gene, then we have a gene networks, and we also have the proteins inside these networks. So what we want to have is our the graph on the left side. We have the cell nodes, and we have the gene nodes, and we also have the proteins nodes. But this kind of graph also have the scalability issue, although we already select only a subset of the gene. So we do a simplification. So we have a decoupled GNN where we first do the propagation and then do the transformation. By transformation, I mean train an MLP. So how we do the propagation is that we first initialize the gene nodes with the observations, with the input matrix. And then we aggregate or propagate the features in the gene networks. And the gene network is completely derived from the string database. And then after we propagate the gene features on the gene networks, we aggregate the gene features to the protein, because we know that there is some mapping between the gene and proteins. And then we aggregate the protein to cell. And these proteins are actually the target protein, that the proteins that we want to predict. So we aggregate those proteins to the cells and use them as a enhanced component. I mean, in addition to the SVD, because we found that SVD is far more. It's too important or useful. So after combining the SVD with our graph-derived features, we notice a significant improvement over the cross-validation score, and this is actually what we find. This is actually our final submission. But before I talk about this final submission, I want to make a conclusion. So basically, our cell feature graph does include extra domain knowledge in contrast to the cell graphs, and also in cell feature graph, we will not be misled by the wrong structures. And we found that this kind of method could be very promising, and we will continue to develop the graph-based methods in the future. Okay, so this is our final submission. It's pretty simple. For the statistic, we concatenate the graph-derived features with SVD, and we use cross-validation. And for multi-omic, we concatenate the gene activity matrix with SVD, and we don't have other ensembles. Lastly, I would like to quickly go through the dance package. We are largely inspired by this series of competitions. So after last year's competitions, we found out, yes, there is some reproducibility issues of the computational methods in single cell analysis, and there is lack of deep learning packages for this community. That's why we start to build up this package called DANCE. And it's a deep learning library and benchmark where we collect 3 modules, the single modality, multimodality, and spatial resolved. And we collect 8 tasks and collect a lot of models and benchmark data sets so that you can run your own models on the benchmark data set easily or apply the sorter models to your own data sets. And we unify the implementation of all the models. So here's a demo of the package where we, collect some benchmark data sets and collect some models and evaluate them. And it's very user-friendly and easy to use. And we welcome everyone to take a look. So this is the QR code. That's the end of my presentation.
Awesome, great, thank you so much. It's actually a very fascinating approach you have here. Can you just clarify, what were the cell activity scores that you mentioned before? How do you calculate? I haven't heard that before. What is that?
I think it's derived from like the GS1 and S2, if I remember
correctly. I think it's like cell cycle scores.
Yes, but it's derived from it. It's not just a cell cycle.
Did you write up a Kaggle discussion post on this approach?
No, we haven't. Sorry.
Okay, it would be very fascinating to read through that if you did.
Yeah, because we're going to continue to polish our graph-based idea, and eventually we'll definitely publish it somewhere.
Absolutely. Great. Well, thank you so much. We're going to move on to our last presentation of the day. So for those of you who've been sort of following the competition, I think Alexander Chervov the name should not be new to you. He's been super active in the discussions and more than happy to have him sort of just talk about his experience in the competition. Thank you so much.
Thank you very much. Let me share the screen. So do you see my screen? Okay, so many thanks to the organizers of the competition for creating such a wonderful event, and for Kaggle staff and for all the participants who created such a wonderful atmosphere. So as we discussed with the organizers, I will more talk not about the data science part, but about some kind of research and making some kind of research activity around this Kaggle competition. So let me switch to the full screen mode, and so it will be a little bit unusual. So let me first introduce myself. So I did a PhD in pure mathematics. Then I was doing research in pure mathematics for 10 years, and I switched to industry. And then I turned several years ago again to the academia, and now I'm bioinformatician and data scientist in the Institute Curie at Paris, France. So I am quite a big enthusiast of Kaggle, but not about the competition side. So I have lots of friends who are masters and grandmasters of Kaggle, and I always hear what they say. But for me, Kaggle is a kind of great thing for learning things and for knowledge sharing. And so the idea of my talk, if some bioinformaticians are hearing me, so to attract them to use the Kaggle, not only for the competitions, but for many other things. So the part one, so Kaggle is great not only for even without competitions. I mean that it's a kind of a cloud service like a Google Colab, but it's much better because you can store data, you can store code, and you can execute code at one place, even with the GPU and TPU. You can organize a teamwork, and you store discussions, ideas, and you make publicly available all what you can do. And so, and you can get feedback from the others. I think this is a kind of exceptional. We do not have another platform and which offers you all these possibilities. Yes, and so as a kind of a joking man, so I forbid you. Let us imagine this guy and his last. Let us forbid something like Google Colab. So I mean, if you see your friends, colleagues, bioinformaticians are working on Google Colab or Jupyter notebooks, come to them and say, please stop using it, please use Kaggle. Why? It's not only convenient, but the main point is in sharing and sharing with the community. So if you put something on Kaggle, it's very easy to reproduce your result. There is a special button, copy and edit. And you will work with the same framework, with the same data, and with the same notebook. I think it is important because of course we can store code on GitHub, then we can put data on, for example, Java, but then for the users, there is a problem. For example, yes, I need to take this data from there, and typically there are small mismatches. The code on GitHub corresponds to other version of the data, and this is a kind of nightmare, I think. And so if you put everything on Kaggle, there will be no such a problem. So if you see some bioinformatician who is working on Google Colab, please ask him to switch on Kaggle instead. So there are on Kaggle, we have already really something like 1,000 data sets related to biology and bioinformatics, thousands notebooks and so on. This is a post which I made about maybe 2 years ago. And what is missing, I think, for the Kaggle, this is something like crystallizing community of the people who are kind of also enthusiasts of the Kaggle, data science, and applications of the bioinformatics. So if you have some contact with the Kaggle administrations, please ask them to create something what is called community for bioinformatics. That means on their forum, they have a kind of sub-forums related to natural language processing, to computer vision, and so on. If they create this one for bioinformatics, I think it would be quite beneficial for the community because enthusiasts can kind of gather there, can discuss, and can create some kind of a joint activity. I think it would be quite useful. So if you have contacts with the Kaggle administration, please consider discussing with them there. So another thing which I want to share with researchers who are not familiar with Kaggle is the following. So the main secret power of Kaggle is actually a community. So Kaggle, it's not only the individuals which are solving the task by themselves, which is of course the most important thing. But on the other hand, Kaggle has a good motivation system for sharing the knowledge. Yes, if you share good code, people will upvote it, and then you can become a kind of Kaggle master or grandmaster. And these titles, they are appreciated in the real world data science community. So if you're kind of a Kaggle master, not only in competitions, this is kind of appreciated. And so you can find a better job and so on. And so people have a good motivation on Kaggle to share their knowledge, share the code, and so on and so forth. So, for example, yes, to give an example of that. So if you have something like a 10 kaglers on Kaggle, I think it will be more efficient than 100 individual data scientists. Why? For example, when you start your work, you need to do explanatory data analysis. You should provide a baseline for solution, but you should look for some literature, what people are doing on this task. On Kaggle, the community will do it for you. So you should just follow the forum. On the forum will appear some good ideas, or in the public notebooks will appear very fast good explanatory data analysis. Very fast will appear some good baselines, and it will be very easy to find them later. Just you go to the code and look for the most upvoted notebooks, and you will see, for example, for this competition, we see by the Ambrose explanatory data analysis, some baseline, some usual, and quite good solution. It's easy to find. And even if people don't share anything, for example, we look on the leaderboard. Who was on the leaderboard at some point? What we can guess from that? We can guess that the gradient boosting are quite good for this competition. Why? Because we know these people who are famous Kaggle grandmasters are quite kind of like gradient boosting. And so even from their position on the leaderboard, we can guess what methods are good for this task. So that's kind of an idea that Kaggle is a kind of community efforts when we solve the task. And so that is important, I think, for the organizers of the competition. So the kind of, I think Kaggle is the best platform to organize the competitions, and maybe don't try to organize them on the other platform, because one of the reasons is quite simple, that it's the most famous, but another thing is about this kind of community stuff. So if you organize it on the Kaggle, then you'll see lots of notebooks produced, lots of discussions, and that will be stored, and that will be absorbed by the community, and that will be used following competitions and the real life experience. I don't think that there are other platforms which allow you the same thing. So that was the first part of my talk, which was kind of, we can use Kaggle not only for competition, but it's just a great platform for knowledge sharing. So the second part is about something like, so we have a competition. So what can we do? Actually, we can organize a kind of crowdsourcing activity, research activity. Why that might work? Let me discuss it. So I think there are two reasons for that. One reason is that actually to succeed in Kaggle, people spend lots of effort. Sometimes people say maybe 100 hours or 300 hours of hard work, but not always their efforts will be, they will have something like a golden medal or something like that, because it's not predictable. You can have a shake up and you can go down to the 100 positions, and it can happen with everybody. So, and all your efforts will be wasted. And that's, I think, a problem for many Kaggler. So if, as a researchers, we say, guys, okay, you spend so much effort, you are so efficient at this task. So, but okay, let's kind of collaborate on something, doing research together, and just spend a small amount of your time, your efforts on this, and do Kaggle as the main thing. Maybe we can. I think many people can agree on this. At least it was an idea. Another idea that Kaggle is already a kind of a well-established brand. So many people want to participate in Kaggle, but they don't know how to start. They need something like a guidance or something like a colleagues or companies, and thus we can kind of attract them via, for example, the social networks. If there is a competition, we can say, okay, there is a nice chance to start something like on Kaggle and also do a kind of research. So as an outcome, the second idea worked better for us. So what we have done? So first we made some advertisement at the social network. It was seen, as far as I understand, by about 20,000 views on this. So we had something like a poll where people can say, yes, we are interested in this. And so about 244 voted yes. So we gave for them some kind of a very simple task. You need to create a notebook, you need to kind of submit, and okay, only 40 passed this simple task. But after that, okay, we can kind of organize the kind of activity on the Kaggle. And so Kaggle is very great for the teamwork. So, for example, now let me show you something like what we are doing now. The competition already finished, but we still want to kind of make a kind of research project around that and maybe write some paper. So we created a Kaggle data set where we can store all the code. We have some discussions when we also we have chat where short discussions, and some the Google Sheet where we share the task, who will do what, and that's all. Very kind of pretty simple team management. So okay, let me skip this maybe. And so I think the idea of this, I think it's a quite nice, quite good idea that we can organize some activities with the focus on the research on Kaggle. Now let me a little bit go to the competition part, to the data science part. Some mentioned our approaches maybe very briefly. So our idea was to create many features which were biologically motivated by some use of the pathways, protein interaction networks, and so on and so forth. So we create maybe some about 100,000 of the features. Then we have some kind of a feature selection, forward selection by the very fast, quick, rich model. And so we can select the features by batches. To construct strong, so that was a kind of feature selection, and we come to this maybe 2,000 features, something like that. Then we can construct something more stronger models or the boosting, so neural networks based on these features, and then blend them. So maybe it's not that much interest in that part. Now, let me just give some example of what we are trying to do now. So if you look on the results of the models, we can see that some proteins are quite good predictable. So this is a bit unexpected from the biology, because we know that there is a very strong post-transcriptional regulation. And so typically the protein level and the RNA level are not highly correlated. And so that results that we can predict some proteins quite good. This seems to be quite interesting. And so I think we need to explore it in future a little bit better. So that's one of the tasks which we want to understand. And so biologically speaking, we want to understand, can we say something about the post-transcriptional regulation? What are the post-transcriptional regulators of these proteins which are predicted quite well? This is one of the questions. Another question that, okay, we have so many machine learning methods, and so we can maybe shed light on some regulation mechanism of the gene. For example, we can look on the teaching importances. And then, for example, we can look on the targets which share many important features. And I think this should mean something biologically. And here is some small examples. So we took, for example, these several genes which are most highly predictable. But the interesting part, which biologically unexpected, all of them are related to one transcription factor, which was not from the beginning. We didn't start from any kind of transcription factor, but we finished with this GATA1 and KLF, which are famous transcription factors, related to the erythroid lineage. I think so. We can find many interesting biological things here. So let me thank my teammates, and my the Curie bosses, Andrei Zinoyev, Manuel Barillo, and Loredana Martinghetti, and all the team members who that other way participated in our activity. And so thank you very much.
Wonderful. Thank you so much, Alexander. I think that was the most compelling advertisement for using Kaggle I've ever heard. So that's also something I think we noticed that was like a big sort of shock for us, was how much incentive there was for people to help with exploratory analysis, with getting quick start notebooks up. When we first started talking to the Kaggle, the Kaggle organizers, and they were saying, they were like, oh, do we need to like provide quick start notebooks and like starter kits and stuff? And they're like, oh no, you don't need to do that. Like the Kagglers will do that for you. We were like, we spent a lot of time last year creating that content. And I think also we were surprised that we spend a lot of time thinking about what pre-processing to do. And it turns out that the best answer is really none. It wasn't worth doing a lot of that normalization, I think, because as we heard today, that's like a huge part of the work that the community is focused on. And I think we are very interested to know, like, what are I didn't quite get many strong trends. Everyone seems to have different approaches.
Yes.
So it seems like there's a lot of different things that work well. So thank you so much.
Thank you very much.
Let's see, are there any other like final questions or comments? I don't want to like hold people up too much past the stop time. This has been an incredibly, wonderful event to bring everyone together. I think maybe the last thing I'll just say is that we're very excited to continue to work with everyone here, everyone who participated in this year's competition. There's the survey that we encourage people who haven't filled out to take. And we definitely want to be able to keep in touch. We're going to be continuing hosting competitions and events and hackathons. And so really hope that some of the folks who were so involved with this competition are still interested in working with single cell data. And actually, I guess there's one question we didn't ask in the survey I'd be interested to know if anyone here has a quick answer to, but a lot of people mentioned the timeline being a challenge. And so we've been doing 3 months for the competitions. Alexander, maybe you have a comment on this, but like, do you think that we should do longer? Like if we did 6 months, is that something you think people would actually take advantage of, or would they do what I do with homework and just wait until, the week before?
I have a proposal on this. I think it would be much better if you split the competition into 2 parts. One competition is for SciSec, another for the multi-ohm part. Because for example, you start with SciSec, which is more easy, and later, for example, 1 month later or 2 months later, you start with the multi-ohm. Because you see, one of the remarks, the number of participants was great, it's about 1,000, but actually expected number of participants would be 4,000, because you can look on the other competitions like Amex or Mechanism of Action. So typically tabular data competition might attract more people. That's I don't know why, but one of the reasons probably it was more kind of complicated for the people to get understanding all this data, especially multi-ohm. And so maybe if you just start from the kind of more simple part and then add more complicated people, okay, if I started this, then yeah.
That's definitely a good, that was something else that we've been talking about too. It's just that people do seem to have taken very different approaches to the two different sets of modalities. Even though in the single cell, so for, in the single cell literature, that's not the case. Like in the published method so far, a lot of people have a single model architecture, and then they talk about applying it across many different kinds of modalities. But that's not the case here. J Serpentine site-seek weight. Yeah, so it is correct that the site-seek prediction was weighted more heavily in the evaluation per. And I think, this is something we discussed a lot. This is our first time organizing a competition on Kaggle, and I think this is actually also the first single cell competition on the platform. And so I think we've learned a lot engaging with the community this year. And I think that this question around weighting is something we want to absolutely be more, pay more attention to for our next competition. This is also a good opportunity if anyone has anything else they want to give feedback to us about the competition or thoughts for next year. But otherwise, happy to stay on for a little bit. Thank you all. All right, great. Well, if that's it, then we'll sign off here and take care, everyone. We'll hope to see you on Kaggle soon. Bye.
Bye-bye.